Welcome you all here in CEC Gurukul online lecture. Learners, in part 1, we had discussed about the derived lipids, that is terpenes, how terpenes are classified and why terpenes are very important biomolecules and why we call terpenes derived lipids. And we have seen triterpenes, which is squalene. And as we know, squalene is a biochemical or a very important intermediate which is present in liver oils or another fatty acids. And it is a polyunsaturated triterpenes which contains 6 isoprene unit and 30 carbon atom, and it is a very important biochemical precursor of cholesterol and other steroids. And we have seen there squalene is a hydrophobic in nature, and it is only soluble in fats, and it is very important part of our biological process, and it is a precursor of cholesterol and other important steroids hormones. And here in this lecture, we will discuss another important terpenes, that is carotenoids. Learners, carotenoid is also terpenes, and carotenoids are tetraterpenoids. And these carotenoids are also comes into the class of lipids. They are also lipids. They are also derived lipids because they are also soluble in fats. They are associated with fat. They are bind with the fats. And yellow, red, orange pigment, which is produced by plants and algae, bacteria, and fungi, represent having a carotenoids. Similarly, these carotenoids gives characteristics colors to pumpkin, carrots, corns, tomatoes, canaries, flamingos, shrimps, etc. And 1100 known carotenoids, that is also called xanthophylls. Carotenoids, which contain oxygen, and carotene, which are purely hydrocarbons. So that gives a specific colors. These are the specific colored pigments which gives characteristics color to the different types of fruits and vegetables. Learners, this is the structure of carotenoids, carotene. So it is a conjugated system of carotenoids which is having 11 double bonds, and this carotenoid which are tetraterpenes, it contains 8 isoprene units. So total number of isoprene unit is in carotenoids is 8, and this is also called beta carotenoids, and that beta carotenes found in different colors: yellow, orange, red colors. And learners, this beta carotenes, which is present as a pigment in colored foods and vegetables, convert in our body into vitamin A or retinols, and that retinol is precursor of vitamin A, and vitamin A is a powerful antioxidant. So vitamin A is biosynthesized from the beta carotene, which beta carotene are the terpenes, are the tetraterpenes, or these terpenes are also lipids because they are dissolved in fats. So this is the structure of aprocarotenol. You can see on this slide the structure of carotenoids, that is aprocarotenol, and it is found in citrus fruit. Similarly, lycopene, you have seen the red color of tomato and red color of watermelon due to presence of that carotenoid, that is lycopene. So this lycopene, which is a carotenoid, which is in red in color, and due to that pigment, we can see the color of watermelon and tomato is red. Similarly, another type of pigment, another type of carotenoid which is present in corn, and we can see the yellow color of corn due to presence of xanthin. So xanthin is another carotenoid, and all these carotenoid contain 8 isoprene units. They are also terpenes, but they are tetraterpenes. So the yellow color of corn is due to presence of a carotenoid pigment, that is zeaxanthin. Similarly, the color of yellow color or slightly orange color of pumpkin is due to presence of violexanthin. So due to that violexanthin, we can see colored pumpkin. Color of pumpkin is yellow. Similarly, cap this is another pigment, capsorubin. So the capsorubin is a red color of red pepper, and this is also carotenoids. So these are the some important pigments which are carotenoids, which are tetraterpenes, and which are lipids because these are soluble in fats, and the color of these fruits and vegetables is due to presence of that tetraterpenes or called carotenoids. Similarly, this is anthocyanin. Anthocyanin is a type of carotenoid. It is a type of pigment which is present in the feather of flamingo. So the shiny color of feathers of flamingo is due to xanthocyanin. Anthocyanin. Now, how terpenes are biosynthesized? We will see. Terpenes are 5 carbon compound, and which is used for the biosynthesis of terpenes. That 5 carbon compound, which is isoprene unit, that is 3-methyl 3-butenyl. So that 3-methyl 3-butenyl pyrophosphate called isopentyl pyrophosphate. So biosynthesis, during the biosynthesis of terpenes, that isoprene unit change to isopentyl pyrophosphate. So this is the structure on your screen, you can see this is the structure of isopentyl pyrophosphate, and that isoprene unit having two phosphate group, two pyrophosphate group. So it is a isoprenoid. This isopentyl pyrophosphate represented as a IPP, and it is a isoprenoid which is precursor and formed from acetyl coenzyme A via mevalonate pathway. So this mevalonate pathway is very important for the biosynthesis of terpenes or squalene and also cholesterols. So here in this mevalonate pathway, the first step is Claisen condensation. So Claisen condensation reactions takes place in the first step of the biosynthesis of fatty acid, except that acetyl and malonyl group remains attached to the coenzyme A. As you can see on the your slide on your screen, acetyl coenzyme A and malonyl coenzyme A, Claisen condensation reaction takes place, and we get acetoacetyl coenzyme A. So in acetoacetyl coenzyme A, only malonyl groups remains attached to coenzyme A. And in the second step, aldol condensation takes place between acetoacetyl coenzyme A and malonyl coenzyme A, and during that we get thioester. So this is the hydroxymethyl glutaryl. coenzyme A. So this HMG, in short form, we can represent here HMG, hydroxymethyl glutaryl coenzyme A. This hydroxymethyl glutaryl coenzyme A is a very important intermediate in mevalonate pathway, and this HMG coenzyme A is needed to make cholesterol. So this is very important biomolecule which is used for the synthesis of cholesterol. So it is a very important intermediate, hydroxymethyl glutarate. So it is used for the biosynthesis of cholesterols. So this HMG reduces to two equivalents of NADPH, which is converted to mevalonic acids. So in the structure, in the slides, you can see here MVA, which is a mevalonic acid or mevalonate, and this mevalonic acid is a key molecule in the synthesis of, in the biosynthesis of or biochemistry of hydroxyl fatty acid cholesterol productions. So overall this is mevalonyl phosphate. So you can see in the slide mevalonic acid in presence of here ADP provide phosphorylation takes place, and during phosphorylation here this phosphate group added to the mevalonic acid and it convert into mevalonyl phosphate. So this is the mevalonate having a phosphate group. Phosphorylation process takes place with the ATP, and with ATP here pyrophosphate group added. You can see on the screen mevalonate, pyrophosphate, and again here decarboxylation takes place, and along with that loss of OH group takes place from this mevalonate pyrophosphate. It convert into isopentyl pyrophosphate, and the isopentyl pyrophosphate changed into convert into dimethyl allyl pyrophosphate, so DMAPP. This is the structure on your screen. You can see here isopentyl pyrophosphate. This isopentyl pyrophosphate in presence of isopentyl isomerase change into dimethyl allyl pyrophosphate. So learners, this dimethyl allyl pyrophosphate used to make isoprenoid, and which is a diverse class of over 30,000 of biomolecules like cholesterol, vitamin K, coenzyme, and all steroids hormones. So here this mevalonate pathway, which is a precursor of steroids. This is the structure of isopentyl pyrophosphate having isoprene unit, which is a terpenes, and phosphorylation takes place over here with ATP, and it added to pyrophosphate group to the isopentyl groups. So the mechanism of converting or mechanisms for converting mevalonic acid into mevalonyl phosphate is SN2 type reactions with adenosyl pyrophosphate act as a living group. And here ATP is an excellent phosphorylating reagent for nucleophiles because its phosphoanhydride bond are easily broken. And this is the slide you can see here mevalonic acid with ATP convert into mevalonyl pyrophosphate with ADP. So this is a very important pathway, and it is a precursor of the biosynthesis of cholesterols. So this mevalonic acid is a precursor in the biosynthesis of terpenes and steroids. Both are the terpenes. As I told you, terpenes are lipids. Similarly, steroids are also lipids. Steroids are also soluble in fats only. So the function of mevalonate pathway is the production of cholesterol. So this is very important pathway for the biosynthesis of cholesterol in our biological process. So here isoprenoid lipid, which is farnesyl diphosphate and geranyl diphosphate. And learners, statins, we have heard about the statins. So statins are the anti lipidemic drugs. So these are the very important drugs that inhibits HMG coenzyme A. As I told you in the previous slides, HMG is a hydroxy methyl glutaryl coenzyme A. So statins are the drugs which inhibits the HMG coenzyme A and reductase in upstream part of mevalonate pathway and reduce the cholesterol level in the bloods. So these are the very important drugs, and these by using these drugs during the process of mevalonate pathway, it reduces the level of cholesterol in the bloods. So biosynthesis of squalene, how the squalenes are biosynthesized. As I told you, squalene is a triterpenes, and it is a 13 carbon isoprenoid compound, intermediate metabolite in synthesis of cholesterols. And you can see on your screen it is having 6 isoprene units, and the structure is molecular formula is C30, C50, C30 and H50 at 2, 6, 10, 14, 18, and 22 positions. There is a double bond at 2, 6, 10, 14, 18, and 22 positions, and with all E configurations. And it is a biosynthesized by coupling of two molecules of farnesyl pyrophosphate. So this is the two molecules of farnesyl pyrophosphate, and that molecules combined to form a squalene. So this is the structure of squalene. And squalene, during the reaction, it is catalyzed by squalene synthase, and that squalene synthase activate the reaction, catalyze the reactions, and two molecules of farnesyl pyrophosphate forms. In a tail to tail linkage takes place here. So tail to tail linkage, squalene is precursor of cholesterol, and cholesterol is precursor of all steroids. So, learners, this farnesyl pyrophosphate react with another molecule of isopentyl pyrophosphate to form GGP, that is garnyl pyrophosphate, which is a 20 carbon compound. That GGPP is 20 carbon compound, and two molecules comes together to form phytoene. That phytoene is a 40 carbon compound, and phytoene is a precursor of carotenoids, which are the tetraterpenes. And these carotenoids are the pigments of plants. So in this lecture we had discussed about the carotenoids, how carotenoids biosynthesized, and also how the biosynthesis of squalene takes place, and how the these squalenes is biochemical intermediate, and activate the reactions and used in the biosynthesis of cholesterol and all other steroids. Thank you.